Om Swastiastu. Hello everyone. We are from SGD B5 and we're going to present our student project entitled The Role of CDKs in Cell Cycles. My name is Bernard Karel Quaresma Fatti, with the last 3 digit of my student number is 103 as moderator for this presentation. These are the names of all the members in SGD B5 with their student number. Now we're going to start with the background of our student project.
Good morning, my name is Gabriela with last 3 digits number 118. The body of our organisms is composed of functional and structural units of life called cells. Structurally, a cell is commonly defined as the smallest unit of organisms consists of matrix of protoplasm and cell organelles. Functionally, cells play a role in carrying out life functions. Cells derive from stem cells that undergo a moderate evolution, which these changes cause these cells to be divided into prokaryotic and eukaryotic cells. Eukaryotic cells are organized by cytoskeleton and its internal membrane consists of DNA, also the cell organelles. Eukaryotic cells reproduce by cell division, which is termed as cell cycle. The cell cycle consolidates 2 principal phases, namely the interphase and the mitotic phase. During the interphase, cells undergo a growth process in 3 phases, which are first gap synthesis of DNA and also the second gap, followed by the mitotic phase consisting of prophase, metaphase, anaphase, and telophase. The eukaryotic cells needs to be controlled and regulated, and this is the responsibility of cyclin-dependent kinases, which are heterodimeric protein kinases consisting of 2 subunits, namely cyclin and CDK as the catalytic subunit. CDKs control the cell cycle by modulating the phosphorylation states of proteins involved in activating specific events in the eukaryotic cell cycle. There are different types of CDK, which are CDK 1 till 4 and CDK 6, along with their associated cyclins being cyclin A, B, D, and E. That's all from me. Thank you.
Greetings, my name is Justin with student number 106, and today I'll be explaining about our problem formulation, our purpose, and our benefits. Alright, so for our problem formulation, first is we need to know what CDKs are. Second, we need to know the role they play in the cell cycle. And third is we need to know the different phases of the cell cycle. Okay, so as for the purpose, we've divided them into 2 categories. Generally, this project is aimed to understand the role of CDKs in the cell cycle, and specifically, the first one is to get a deeper and more in-detailed look as to how CDKs perform in the cell cycle. Second is the mechanisms of CDKs in the cell cycles. And third is to provide a deeper and more rich understanding of the cell cycle. And finally, for our benefits, we've again divided them into 2 categories, theoretical and practical. As for our theoretical benefit, this paper was written in the hopes that it could enrich the knowledge of the readers and serve as future references for studies to come. Specifically regarding to cyclin-dependent kinases and the role they play in the eukaryotic cell cycle. And as for our practical benefit, we hope that by writing this paper, it could provide a deeper insight into CDKs and the role they play in order for future developments in healthcare and medicine. That'll be all from me. Thank you.
Let me introduce myself. My name is Bagus Tresna Indra Putra Pramana with the last 3 digits student number 104. I'll be explaining about the cyclin-dependent kinases in eukaryotic cells. Cyclin-dependent kinases or CDKs are a family of small serine or threonine kinases found in eukaryotes. CDKs are not active in the monomeric form, which require an activating subunit to be active as protein kinases. The process of the cell cycle in eukaryotic cell occurs in a specific sequence and is controlled by the CDKs. The eukaryotic cell cycle control system is based on the S phase, and mitosis is being controlled by the cyclin-dependent kinases or CDKs, complexed with the S phase cyclins or SCDKs and mitotic phase cyclins or MCDKs. Cyclin CDK complexes activate or inhibit hundreds of proteins involved in cell cycle progression by phosphorylating them at specific regulatory sites. Therefore, proper progression through the cell cycle is governed by activation of the appropriate cyclin CDK complex at the appropriate time.
Okay, my name is Husnia Adiba with 3 last digit number 102. Next. Okay, so the CDK is activation. So the CDK is not active in the monomeric form. So they have to bind to a certain protein that can activate them. That is called a cyclin. This cyclin will activate the CDK and CDK will trigger stage in the cell cycle. You can see here that in G1, G1 S phase CDKs activate the transcription of gene required for DNA replication. The S phase CDKs then activate DNA replication and DNA synthesis will commence. Once DNA replication is complete, cells enter the G2. In late G2, mitotic CDKs triggers entry into mitosis. So, how does exactly cyclin bind to the CDK? So, CDKs has active cyclin wherein ATP and substrate bind to it. So, when the cyclin bind to it, they're composed by two domains and each of them has alpha helicase. The first domain will correspond to the cyclin box region, was for the CDK binding and activation in CDK1 and CDK2. The binding causes the alpha helicase to move and conformational chains to a more proactive conformation for catalysis. Aside for stimulating CDK activity, the cyclin subunit, however, can also interact with substrate protein that contributing to the substrate specificity of cyclin CDK complex.
Okay, thank you for the opportunity. My name is Fatti with reality of new number is 116. And now I will explain about CDK inhibitors. CDK inhibitors play a role in controlling CDK activity by inhibit the activation of CDK enzyme. There are two ways to inhibit CDK. First, through inhibitory phosphorylation, which is regulated by tyrosine and threonine, which interferes CDK control in G1 and S phase. Second, through the CDK inhibitors, which bind directly to cyclin CDK complex and inhibit their activity. Divided into two classes, the first contains INK4 family and the second contains CIB family. Next, and then I will explain CDK inhibitors in cancer treatment, divided into two generations. The first compound CDK inhibitors that have poor selectivity and high toxicity. In this generation, they are flavoparidal, used against chronic lymphocytic leukemia cancer, and then roscovitin, used against nasopharyngeal carcinoma, and then UCN, used to against breast and colon cancer. Second generation of these are safer and more effective. In this generation, they are palbociclib and ribociclib, who's used to against breast cancer, and abemaciclib, used to against breast cancer and colorectal cancer. Thank you.
Let me introduce myself. My name is Kevin and my last three-digit number is 105. Right now, I'm going to explain about cyclin-dependent kinase localization and general function in cell cycle. First, there are 20 types of CDK that have been identified that are involved in regulation of cell cycle, transcription, and splicing. The CDK types that are involved in cell cycle are CDK1, 2, 4, and 6, which are located in the nucleus. In cell division, the CDK ensure the replication of DNA in each cell and equally divided in both new cells. CDK1 promote the transition from G2 phase into mitosis. It is also controlled by checkpoint kinase, which ensure that incompletely replicated or damaged DNA is not distributed to the daughter cell. CDK2 is a significant cell cycle component that regulates the G1 to S and S to G2 transitions. CDK2 or cyclin E must phosphorylate RB to generate S phase entry. CDK4 and 6 complexes will trigger the phosphorylations of RB, which allows the separations and transcription of E to F target gene such as cyclin E, that if combined with CDK2 initiate the G to S phase.
Greetings evaluators, my name is Jiraldi with the last three-digit number 119. Different types of CDKs are found to be expressed in particular phases of the cycle, depending on the checkpoints. The constitutive synthesis and destruction of cyclin CDK pairs depend upon various signaling processes and proteins. Based on the figure on the left, the G1 cyclin operates at the early G1 phase, which increases progressively upon the addition of growth factors. In fact, at the transcriptional level, The induction of cyclin D1 by growth factors is dependent on the RAF mitogen activated kinase signaling pathway. Next, the graph trend of G1S cyclins peak at the G1 transition and experiences a big turnover due to the ubiquitination process and proteasomal degradation. CDK2 and cyclin A pairs phosphorylate substrates that initiates DNA replication, and they also plays a role in coordinating the completion of S phase. At last, the cyclin B CDK1 pairs phosphorylate numerous target proteins to trigger fundamental events such as the separation of centrosomes, condensation of chromosomes, breakdown of the nuclear lamina, and so on.
Good morning, my name is Nethania with the last 3 digits of name 112. Cell cycle is a process of cell reproduction. The cell cycle is divided into 2 main processes, which are interphase and mitotic phase, with resting intervals in between. Interphase consisting of G1, S, and G2. The mitotic phase can consist both mitosis and meiosis, depending on the type of cell. In somatic cell types, mitosis will occur, whereas in gamete cells, such as eggs and sperms, meiosis will occur. Mitotic phase is divided into 2 processes, which are karyokinesis and cytokinesis. Karyokinesis is a phase of nuclear division consisting of 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase, where cytokinesis is a phase of cell division.
Hello, my name is Natasya Pramatya Putri with student number 2202511111. So I will tell you about the phase of the cell cycle. Cell cycle divided into interphase and M phase. The interphase consists of G1, S, and G2 phase. G1 phase is for the synthesis of the DNA, the growth of the cell size, and the duplication of organelles. S phase is for replication of the DNA and duplication of the centromeres. G2 phase is for enlargement of the cells and supplying the protein and organelles for M phase. Okay, next is G0 phase. This phase exists between M phase and G1 phase. G0 also known as resting phase. This phase divided into 2 stages. The first one is quiescent stage and the second one is senescent stage. The quiescent stage is where the cell can re-enter the G1 phase, and the senescent stage is where the cell cannot re-enter the G1 phase. Okay, next one is M phase. Mitotic phase occur in somatic cells. Mitotic phase consists of first, prophase, when the DNA condenses become chromosomes. Second one, prometaphase, the nuclear membrane disappears and the microtubule growing from the centromeres. The third is metaphase, when the chromosomes align on the center of the cells. The fourth is anaphase, is when the sister chromatid separates into 2 different poles. And the last one is telophase, when the nuclear membrane reappear. And the next is meiotic phase that occur in gamete cells. Meiotic phase consists of meiosis 1 and meiosis 2. Meiosis 1 begin with prophase 1. In prophase 1, DNA condenses and chromosomes duplicate, become sister chromatids. These sister chromatids join at the centromere. Then the homolog chromosomes undergo synapsis to form a complex of the 2 pairs of sister chromatids. This process is called recombination or crossing over. The next is metaphase 1, when the chromosomes homolog align at the equator. The next is anaphase 1, the chromosomes homolog separate into different poles. The next is telophase 1, when the cell divided into 2 daughter cells. And the next stage is meiotic 1. Meiotic 2. Actually, the meiotic 2 process are the same with mitotic process. Thank you.
Allow me to introduce myself. My name is Tracey Odella Jiraldi Akemah with last digit number 115. Today I will explain about cell cycle progression and control. Cell cycle progression is a tightly regulated process that involves numerous checkpoints prior to cell division to ensure that the outcome of the critical processes are engaged well. Cell cycle is largely driven by the mediation of cyclin-dependent kinases and their regulatory subunits, which are called cyclins. CDKs acts as master controllers in the cell cycle by forming complexes with cyclins to phosphorylate substrates in specific phases. CDK 1, 2, 4, and 6 controls the CDK activity. In the cell cycle, CDK4 and CDK6 binds with cyclin D to promote cell cycle entry. After that, cyclin E activates CDK2 in the late G1 phase, activating the E2F transcription, which progresses the cell to the S phase. In the early S phase, CDK2 binds with the cyclin A, which progresses the cell to the G2. CDK1 binds with the cyclin A in the late G2 phase and initiates the mitotic entry, and at the M phase, CDK1 binds with cyclin B to complete the cell cycle. And cell cycle checkpoints are surveillance mechanisms that monitor major events to prevent genetic errors from happening during the cell division.
Let me introduce myself. My name is Samantha Theobald with name 22025111108. Body of organism is composed of cell, divided into eukaryotic and prokaryotic. Eukaryotic cell reproduce by undergo a cell cycle. Cell cycle is divided into interphase, M phase, and GO phase. Interphase, the preparatory phase of cell cycle, consists of G1, S, and G2 phase. M phase, or cell actual division, consists of prophase, metaphase, anaphase, telophase, and cytokinesis. There's also GO phase, the resting phase. Cell cycle is tightly regulated by numerous checkpoints and act of cyclin-dependent kinase, with cyclin as regulatory subunit. Each type of CDK required in regulating the cell cycle owns a distinct function. In controlling the CDK activities, the CDK inhibitory hold an important role in order to maintain the balance. The clinical implementation of CDK inhibitors are useful for cancer treatment through chemotherapy related to its control function.
So that's the presentation of our student project. Thank you so much for your great attention and have a nice day.